MRC Laboratory of Molecular Biology. She was celebrated last night as the Blavatnik Awards laureate in life sciences. The title of her talk is Decoding the Immortality Enzyme, an Atomic View of Telomerase. Please join me in welcoming Dr. Kelly Nguyen to the stage.
Bây giờ là bài thuyết trình của em Giang.
Good morning, everyone. It's my great honor to be here and to tell you about my research. Research in my lab has been focusing on a large and complex molecule in the cell named telomerase. A major motivation for working with telomerase is the important role in human health. Telomerase upregulation, which occurs when telomerase is produced at levels significantly higher than normal, has been shown to be a hallmark of about 85 to 90% of cancers, whereas this deficiency leads to a range of premature aging disorders. Our goal is to understand how telomerase works and how it is controlled in cells. This fundamental understanding is the first step towards identifying new therapeutic avenues. Before I tell you more about the details of my research, I will first introduce what telomerase does in the cell and why it is important. As you might know, the genetic material for all living organisms are stored in a biological molecule called DNA. In fact, each cell in our body contains about 2 meters of DNA. To fit into a tiny cell, DNA is heavily condensed into what we call chromosomes. Here is what a condensed chromosome looks like. In our work, we're particularly interested in studying the very ends of chromosomes, which are known as telomeres. If we take a closer look at the telomere, you can see that it contains repetitive DNA sequences and linear ends. The role of telomere is to protect the chromosomes. If you imagine our DNA is a shoelace, telomeres will be the plastic tips at the end of the shoelace. If the shoelace loses these tips, it will fray. This is what happens to our chromosome if they lose their telomeres. In an ideal situation, if all the chromosomes have their telomeres, all is going well. However, the majority of cells in our body are constantly dividing. It means that one cell becomes 2, 2 become 4, and so on. As cells divide, telomeres actually get shorter and shorter until they reach a critical point when they become too short. This would then trigger a stop to cell division and eventually cell death. This is where telomerase come into play. To overcome this telomere shortening, telomerase adds telomere back to the chromosome ends to keep them protected. This allows cells to continue dividing and achieve what we call cellular immortality. This is why telomerase is often called the immortality enzyme. To understand how telomerase works, our goal was to visualize it as a molecular level. Despite the critical role of telomerase in human health, it has remained elusive for many years. Telomerase were discovered by Elizabeth Blackburn and Carol Greider in 1895. Since then, there has been a tremendous amount of genetic and biochemical work on this enzyme. 24 years later, Blackburn and Greider, along with Jack Shostak, were awarded the Nobel Prize in Physiology and Medicine for discovering telomerase and its role in protecting chromosomes, illustrating the magnitude of this discovery. However, it took 28 years since the original discovery until we got the first glimpse of the human enzyme. This first structure was generated in 2013. While this structure tells us about what the enzyme looked like roughly, it lacks molecular details. This makes it challenging to understand how telomerase is regulated in the context of disease, which in turn hampers drug development efforts. In 2021, my group published the very first atomic structure of human telomerase, providing the field with unprecedented insight into the function of this enzyme. This structure was published over 30 years since the initial discovery of the enzyme. In addition to generating the very first atomic structure, our other major contribution to the field has focused on streamlining a method to produce telomeres at the scale needed for this type of microscopy. I want to illustrate some of the challenges that my research has addressed to get us to this point. You might have come across a phrase, seeing is believing, and this is true in biology. Visualization of molecular objects is a powerful way to understand how they work. For example, our eyes are very good at seeing objects up to a millimeter scale, such as ants or other insects. However, when things are in the micro and nanometer scales, such as individual human hairs or cell, one will need to use specialized instruments such as microscope to visualize the details. When we get further down to the level of individual proteins like telomerase, we need a specific kind of microscope called an electron microscope. Electron microscope requires a significant amount of purified protein to attain a clear picture. Telomerase is one of the rarest enzymes in the cell. Even in cancer cells, where it's highly expressed, there are still only between 50 to 250 molecules of telomerase per cell, it is also absent in somatic cells, which account for the vast majority of cells in the human body. For these reasons, getting sufficient telomerase for visualization would require a lot of human cells, a few hundred liters of cells to be exact. My group has made significant breakthroughs in our ability to produce telomerase at the scale needed for electron microscopy. To produce telomerase, we used human cells to produce telomerase with a tag, and then we break up the cells to get a mixture of all cellular molecules. Next, we isolate telomerase through functionalized beads that can recognize the tag on telomerase. Removal of telomerase from these beads give us our purified telomerase. This sample is unloaded on an electron microscope. You can think of this microscope as a really powerful magnifying glass, which magnifies telomerase tens of thousands of times to give us this detailed view of the enzyme. Just to give you an idea how small this is, the longest dimension of telomerase is about 30 nanometer. To put this in perspective, the width of a single human hair is approximately 80,000 to 100,000 nanometer. Once we generate the overall structure of telomerase, we use it as a map and fill in all the different individual molecular components. First, I would like to draw your attention to this bottom half of the structure. In blue is telomerase DNA that mimics the end of a chromosome. This structure allows us to understand how telomerase interact with the end of a chromosome. Next, to understand how telomerase is controlled at telomeres, we visualize telomerase bound to protein factors at chromosome ends. Furthermore, we map premature aging disease mutations on our structure as shown by these spheres. This allows to explain why these mutations give rise to telomerase deficiency. Recently, we discovered a rare dimeric state of telomerase in which two telomerase molecules associate with each other. The interface between these two molecules is crucial for telomerase function in cell. When we looked more closely at its interface, we found several mutations that are associated with disease of premature aging. Taken together, our work provides important insights into telomerase function, regulation, and disease mutations. The knowledge we now have places us in a better position to develop strategies for modulating telomerase. One can think of ways to inhibit telomerase to treat cancer or stabilize telomerase for treatment of patients with premature aging syndromes. I want to return to this timeline. Our goal is to use the structure insight to inform therapeutic strategies. It was only recently, in 2024, almost 40 years after the discovery of telomerase, that the FDA approved the first telomerase inhibitor. This inhibitor was developed based on the DNA sequences of telomeres. We are optimistic that our contributions to fundamental telomerase biology will accelerate this timeline and produce new thalamus modulation agents in the next several years. Finally, I want to illuminate an additional avenue of our work. Inspired by the Darwin Tree of Life initiative, which aims to sequence as many species as possible in the UK, we are also exploring telomeres and telomeres in different species to understand the diversity of telomeres and telomeres across evolution. At a time when the global community faces profound challenges from climate change and accelerating species extinctions, this research is especially timely. By uncovering the remarkable diversity of telomere biology across life on Earth, we hope not only to deepen our understanding of evolution, but also to provide the knowledge that might contribute to future conservation efforts. With this, I will end my talk by acknowledging all lab members, past and present, collaborators, colleagues, and facilities at the Institute, all of whom who made this possible. I would like to acknowledge to thank the Blavatnik Foundation and the New York Academy of Sciences for this great honor. Last but not least, I would like to thank all funders who support our work over the years, and I thank you all for your attention.
Rất rất nhiều.